Alright, so we're going to start up now with our second section on the acoelomates and pseudocoelomates. And we're still in our acoelomate section. We're going to start up now with the cnidaria, which are the animals that include things like the jellyfish, the corals, the anemones, and a few other things which you might not be quite as familiar with. So, with the cnidaria, this is another group that, like the sponges, is very much a marine-dominated or saltwater-dominated group. There are a few freshwater species of hydra, as well as even some things that very closely resemble jellyfish, even though they're not really classified that way, that are freshwater, but the vast majority of this group is going to be marine. So this group is diploblastic, meaning that when they're embryos, they have only an ecto and an endoderm. They do not have a mesoderm. They're going to have that mesoglia that we talked about back in our first section. So they're really the only group that we're going to see in this entire course, really of all the animals, that is diploblastic. So they're unique for that reason. They do have some tissues, but they do not have organ level specialization. So they are more specialized than the sponges, but less specialized than really every other group of animals. So they're a unique group. They are diploblastic, they're radially symmetrical, they have tissues but not organs. This is a group which is very much its own thing compared to some other things. So they're going to have, for example, some epithelial tissue, they're going to have reproductive tissue, they're going to have nervous tissue and muscle tissue. That's really kind of about it. They don't really have organ level things for reproduction, circulation, excretion. Most of those processes, with the exception of reproduction, take place through diffusion. So it's a very simple way to get things circulated, pick up oxygen, get rid of CO2, those sorts of things, compared to what other animals are going to have. They don't have a centralized nervous system. They do have nervous tissue, and they actually have what's called a nerve net. So with this kind of a nervous system, what you're going to see is that essentially each part of the body is under its own localized control. There isn't a brain, spinal cord, or even a ganglia, or set of ganglia for that matter. It's really nerves set up throughout the entire animal that control each part of the body independently. So that's kind of what we're going to see with this group. They are very receptive to the environments. They have touch receptors, they have gravity receptors, they have light receptors. There are some that will move away from light. There are some that move toward light. There are some that if you sort of hold them in your hand, they will flip upside down, which we'll talk about. There's a picture of one of those that we'll see here in a little bit. So, they are very responsive to the environment, but they don't really have any sort of sensory organs. They don't have eyes or ears or noses or anything like that, but they do have, like I said, sensory cells or tissues for perceiving various things in the environment. So we mentioned the fact they're marine. Not really too much more to say about that, I suppose. But cnidarians are always going to be predators. They're always carnivorous. They're always predatory. There are no herbivores. There are nothing like that. Not filter feeders. They're actually out there actively catching and consuming prey. The main cells they use to do so are called cnidocytes. Some of the older textbooks you might run into, sometimes you can still find the name nematocyte used. That name really isn't used very much anymore for the cell type. Cnidocytes, though, is a much more common term for the type of cell that's used to capture the prey. And specifically on those cnidocytes, there are what are referred to as nematocysts, which are stinging cells that are used to capture prey. And these are really a unique structure, very fascinating, very, for such a simple animal, a very, very powerful way to capture prey. And we'll spend a little bit of time later on talking about the structure of those nematocysts. Cnidarians, something else kind of interesting about them is they come in two body forms. There is a polyp form, which is what you see in this top picture here. This anemone very nicely illustrates what a polyp form looks like for a cnidarian. It's sort of like a weird looking plant. It's got a stalk, it's got tentacles, it is attached to the bottom, not really moving around. The vast majority of cases they don't move when they're in the polyp form, but they're still able to catch prey with the tentacles they have sort of sticking up into the water here. The other form is going to be the medusa form, and the medusa form is what you think of when you think of what a jellyfish is. So it's got kind of the bell part up here, it's got tentacles hanging out down here, and they're able to move throughout the water. Now, how they move might be a little bit different than what you sort of expect, but they are modal and they are able to move around. In most of the cnidarian groups, these two body forms take turns producing each other. And so you're going to have a polyp form, which gives rise to medusa, and then medusa reproduce sexually. The eventual outcome of that, once they go through a juvenile stage, is going to be another polyp form. So that differs a little bit depending on which group of cnidarians we're looking at, but for the most part, All the groups have these two body forms they use to make their way through their life cycle. So back to the polyp form for a second. A lot of the polyps, especially when we're dealing with the corals, can be colonial. You can have literally millions or hundreds of millions of animals living in one spot, and these are the animals that form, of course, coral reefs. Within a single patch of coral reef, even though you might have a million or 100 million animals in that one patch, it turns out they're all clones of each other. They're all genetically the same thing, and they've reproduced by budding. And if you were to take any animal out of that cluster, you'd find out they have exactly the same DNA because they are clones of each other. And a lot of times too, especially within the corals, you'll see them producing that internal skeleton that we think of when we think of what a coral reef is, the actual physical structure of it. It's kind of like rock. That's the internal skeleton produced by the coral animals as they were going through their life cycle. Now, back when we covered the sponges, we talked about intracellular digestion. Here, we're dealing with internal extracellular digestion. So this is what every other kind of animal is going to do, from the cnidarians all the way up to you. We use extracellular digestion, where food is brought into some sort of a cavity, and enzymes or acids are excreted or secreted to break down those food particles, and then the nutrients are absorbed and distributed throughout the body. Cnidarians invented that. They're the first animals to do this, and every other animal group after them has continued this tradition of extracellular digestion. The sponges are really the only group that uses intracellular digestion, where food is broken down on a cell-by-cell basis. So, this is the group that invented extracellular digestion. Now, we're going to go back and talk about those cnidocytes for a few minutes here. Cnidocytes, remember, are the stinging cells that are used to capture prey. Cnidocytes possess those nematocysts, which are the stingers, or they're really more like a microscopic harpoon, really, that's used to capture prey and sort of make it or incapacitate it. These are found typically on the tentacles of the animal, but they can be found throughout the body elsewhere as well. Essentially, what a nematocyst is like I said, a harpoon. It's attached to a thread, and that thread is able to deliver a poison or a venom to the prey. These nematocyst harpoon-like structures are even recurved, so that once they go into potential prey or even a potential predator, because they're used for defense as well, they're really hard to get out. They have backward-pointing spines that make them very difficult to remove once they've been injected into a prey. This firing of a nematocyst is literally the fastest known cellular process of any organism anywhere. It is a remarkably fast process, so that once the part of the animal is touched by a predator or a prey, essentially what happens is you see a very, very rapid osmotic change, where the inside of the cell becomes extremely hypertonic, which then draws in an immense amount of water, generating high pressure, which then fires out the harpoon, where it can then embed into whatever triggered the firing of that structure. So that's basically the idea here. On this surface of this particular anemone, we have these epithelial cells, and then we have a cnidocyte. And this cnidocyte has this nematocyst, which is unfired. It has a trigger, and when this trigger is touched, that fires the nematocyst out of the cell and into whatever has caused the cell to fire. If it's a prey item, that prey item can be incapacitated. If it's a predator, that predator might very well think twice about further attacking that anemone. These are, depending on what part of the animal you're looking at, very, very abundant. It's not like they have just one or two of these. They will have tens of thousands, if not more, of these nematocysts on even a very, very small surface of the animal. So it's very likely, if you were to touch an anemone, a coral, a hydra, a Portuguese man of war, jellyfish, whatever, you're not going to get stung with just one of them. You're going to get stung with at least several thousand, if not more. So they're very, very abundant. And if you've ever been stung by a jellyfish or Portuguese man of war, very, very serious to deal with. This is sort of an illustration of one. Basically a similar picture we saw on the last slide. This is a scanning electron micrograph of a nematocyst. So this is the structure. These are the hair triggers of other nematocysts. This is the cnidocyte itself. This is the cell that contains it. But you get a really good sense for those barbs, like I said, that point backwards. So once this goes into an animal's body, it's going to be hard to get back out again. And so getting stung by a jellyfish is not a trivial thing, depending on what kind of jellyfish it is, first off, but also your degree of sensitivity. Something like this upside-down jellyfish, that's actually its name, very, very minor. You really don't even feel anything when they sting you. You can handle them, you can move them in your hands. They don't really feel like anything. This box jelly up here, on the other hand, box jellies have one of the most powerful stings, and really, their venom is on par with the venom of almost any other snake, insect, millipede, or not millipede, centipede, almost anything else you might find on land that could cause fairly severe pain. These can actually be fatal if you're stung by them in the water. Portuguese man of war, kind of similar, not quite as potent, but they're much bigger than box jellyfish. These things can be 25 feet long when you include the tentacles. And the kind of the cool thing about these too is they're not just one organism; they're actually a collection of organisms. They're colonial, so this looks like one big kind of pinkish purple jellyfish. It's actually thousands and thousands and thousands of animals that are all kind of clustered together to form the structure which you see in front of you. But with the tentacles and the bell combined, this thing, like I said, could be 25 or 30 feet long. So if you get tangled up in one, you could have a really bad day, and they can be very, very painful if you get stung by a Portuguese man of war. We talked about the body forms before, so we have our polyp form down here with our tentacles, the mouth, the gastrovascular cavity, which is where really all the basic processes take place, from gas exchange, respiratory gas exchange, to digestion, to even gametes released during reproduction. They all come out from the gastrovascular cavity. Then the body stalk eventually down here as well. The medusa is the other form, which also has the gastrovascular cavity, as well as a mouth. The main part of the jellyfish is called a bell, and then tentacles hanging out down here. All right, so that's a pretty common body plan for a cnidarian. We talked about the gastrovascular cavity. Like I mentioned, a lot of the functions of the animal take place here. Gas exchange, digestion, waste disposal. These guys use ammonia as nitrogenous waste. They get rid of it through their gastrovascular cavity. A lot of them will also form their gametes and expel them that way as well through the gastrovascular cavity. Sorry about the noise. They have two layers to the body wall. They have an epidermis, which is the outer layer, and also the layer that lines the gastrovascular cavity on the inside. And then they also have the gastrodermis, which is everything between the two epidermal layers. So this is largely what's composed of mesoglia. And mesoglia, like I mentioned earlier, is a jelly-like substance. It's like 90 or 95% water. So the body of a jellyfish, the vast majority of it is based on water and really not much in the way of solid tissue. Now, we have not yet talked about skeletons in animals, different kinds of skeletons. So cnidarians use what's called a hydrostatic skeleton. And essentially what this is fluid enclosed within the tissues of the animal. And so if there's enough fluid within those tissues, it generates a rigid structure and helps give the animal its shape. Fluid under pressure generates structural integrity. This is the whole basis behind how hydraulic equipment works. This is the basis behind how a lot of animals' skeletons work. Their skeleton is not made of bone or chitin or any sort of hard structure. It's actually based on water enclosed within a space and the force that fluid exerts back in the opposite direction. Some of these animals can build a more structurally based endoskeleton, like we mentioned before. With the corals especially, they build the reef that's based on calcium carbonate, which is limestone. Sometimes they can use chitin as well, but usually it's calcium carbonate. But the vast majority of the cnidarians, from the jellyfish to the hydrozoans to the cubozoans, which are the box jellies, all of those guys use a hydrostatic skeleton, with only the corals being the ones that sometimes use an internal skeleton made of chitin or calcium carbonate. As far as body forms, now the cnidarian life cycle is a bit more complex than we talked about with the sponges. The typical thing for most cnidarians, if we sort of pick any point in the life cycle and start, let's start with the zygote. Right? So a zygote is a diploid form. It's a single cell. A zygote will go through mitosis, and in the cnidarian world, that zygote will eventually produce a polyp. So the stationary form, which looks kind of like that weird upside-down plant kind of a thing. Now the polyp form, although it's stationary, will eventually start producing, again, diploid, medusas through asexual fragmentation. So they'll actually start giving off or releasing medusas that are clones of that polyp genetically, but a different body form, to go off into the water. Now the medusas are free-living, and they can swim, and so eventually they're going to start producing gametes through meiosis. So these medusas that you see, when you think of a jellyfish, they're either male or female. All right, there's a term for that we'll talk about here in a second. And they're going to start producing gametes, sperm cells and egg cells, through meiosis. Now, eventually, those gametes might meet up, fertilization might occur, and when fertilization occurs, that brings us back to our zygote stage. All right, so it goes zygote, polyp, medusa, and then eventually back to zygote stage. Now, there's one other kind of intervening stage that you'll see there in some of these things after the zygote, but pretty quickly that's going to turn back into the polyp. So we're not really going to emphasize that too much quite yet, but we will here shortly. Like I said before, most of the cnidarians have both body forms in their life cycle, both polyp and medusa, but it's only the medusa that does the sexual reproduction. So for the corals and the anemones, for example, that only have polyps, they really don't do much in the way of sexual reproduction. They're primarily asexual, although they can become sexual for short periods. Now, the idea of having separate male and female sexes in a species is referred to as being dioecious. Right? Dioecious, because I guess I don't have it on this slide, is spelled Diocious, dioecious. Dioecious means separate male and female sexes. Right? So humans are a dioecious species. You are either male or female. You're not both. And if that does occur, that's not kind of like a typical thing for our species. So dioecious is kind of the norm for humans and the norm for most cnidarians as well. You can also refer to a dioecious species as being gonochoric. Gonochorism is a state of being either male or female, but not both at any given point in time. All right, so with that, there is also an alternative. You can also have animals which are monoecious. Now, monoecious is where individuals are both male and female at the same time. And so we're going to see that with our next phylum that we cover with the flatworms. We're going to see it here and there in a few other groups like the annelids, the segmented worms. Monoecious is male and female at the same time. So if you translate those terms, the term monoecious literally means one house, where essentially you have both males and females under one roof, so to speak, one individual. Dioecious means two houses. So individuals are either male or female, and they occur on, like I said, different specimens, different individuals, if they're dioecious. So cnidarians are dioecious. Now, the other stage that some cnidarians show is called a planula. And a planula is a juvenile stage that only occurs in the groups that have the medusa form. So we can add one extra step into our life cycle. Remember, we started with a zygote. Zygote, in this case, becomes planula. Planula settles down to form a polyp. Polyp produces medusas. Medusas produce gametes, sperm cells and egg cells. Gametes then form the next generation zygote. So a planula is a stage between the zygote and the polyp in these organisms. It's essentially a very, very short-lived but free-living stage of some cnidarians that eventually turns into the polyp form. Eventually, like I said, it turns into a polyp, and the polyp then produces other medusa, if it's a group that produces medusa, or it produces other polyps asexually in the case of things like corals and sometimes anemones. So those are our planular larvae, and this slide actually has a visual of a fairly typical cnidarian life cycle. We started off with our zygote or fertilized egg. We had a planula, which swam around for a little while, became part of the plankton, and then formed the beginning of the polyp stage here. The polyp stage has, in this case, both feeding polyps, which have tentacles, and reproductive polyps, which have little baby medusas inside of them. The baby medusas are then released. They grow up and eventually start producing gametes, and the gametes revert back to the zygote stage eventually once they're fused, once they fertilize. So that's a pretty typical cnidarian life cycle for most, but not all, of the cnidarians. So with that, some things they don't have, obviously we said organs. They don't have any sort of circulatory system at all. Everything happens through diffusion, so whether it's waste, whether it's nutrients, whether it's oxygen or CO2, it's all moving throughout the body of the animal through diffusion. So what that means for an animal that uses diffusion to circulate nutrients and waste, number one, they can't be very big, because the bigger they are, the farther those things would have to move, and the longer it would take, and so therefore, they wouldn't get the stuff they need where they need to get it as quickly. The other thing that it means, and this is kind of related, is that they're not going to be as metabolically active, because things that are really metabolically active use a lot of oxygen, they make a lot of CO2, they use a lot of nutrients, they make a lot of waste. And so for something like a bird or a mammal, kind of the peak of metabolic activity, we have to have a circulatory system to move all that stuff around in our body fast enough to support us as a very active organism. Things like cnidarians, they don't use a lot of nutrients. They don't use a lot of oxygen. They don't produce a lot of waste or CO2. So diffusion is enough for them to meet their needs. For us, no way. We're too big. We're too active. We have to have a circulatory system. And really most animals are that way, where they have to have some form of circulatory system. related to that as well, respiratory systems, right? So no way to actively bring in oxygen or get rid of CO2. It's all based on diffusion, which again, diffusion does not support a very rapid metabolic rate for a really big animal. So you know they're not going to be huge, and you know they're not going to be super active metabolically. They also don't have any sort of excretory structure, so nothing like a kidney or anything similar to that we're going to see in any other animal groups. They don't have those either. So what we're going to do next, we're going to actually cut out this section now, and when we come back for our third section of this chapter, we're going to talk about some details of the 4 classes of cnidarians, talk about what they are, what they look like, how they make it through their life cycle, and then pretty quickly after that, we're going to move into our discussion of the flatworms.